This is the story of a boy, a pea, a pig, and a lot of bacteria. The boy is James, and he's just like any 14-year-old turning up for another day at school.
There is a certain amount of...
James is in year 10, studying for GCSEs and hoping to do well enough to go on to A-levels and university. He wants to be a geologist. He likes sports. Basketball is his favourite. And he enjoys weight training. It's only at lunchtime that there is anything different about James's day at school. As well as his packed lunch, he has to carry with him this. It's a hypodermic needle, which he uses to give himself an injection. James is diabetic. He has to do this 4 times a day. Though James looks like a healthy young boy, his body doesn't quite work in the way it should. It doesn't produce enough insulin. Insulin is a very important hormone. It's part of the body's control system. Insulin enables the cells of the body to absorb sugars that build up in our blood after we eat foods such as bread or potatoes. Some people like James don't produce enough insulin to keep their blood sugar at the right level. So he has to put insulin into his body by way of an injection to make up for that. The remarkable part of this story is where the insulin for James's injection comes from. The traditional source is the body of an animal. Today, it comes from here. These are bacteria, which have been changed by genetic engineering so that they produce insulin. They don't do it naturally. When they reproduce, the information about how to make insulin is passed on to the next generation of bacteria, and to the next, and so on. The end result is big business. Lots of bacteria producing lots of insulin to be used by people like James. But how is that information about insulin production passed on? All living things are made up of cells. Within most cells, there is a nucleus, and within the nucleus, there are usually several pairs of chromosomes. Different species have different numbers. The chromosomes consist of long threads of a chemical called deoxyribonucleic acid, or DNA for short. A thread of DNA is so small that it can't be studied in detail with an ordinary microscope. This is a computer simulation of DNA, which scientists use to examine its structure. The DNA is actually 2 parallel strands of molecules, linked together by cross pieces, and it's twisted into a spiral. The wireframe model can be changed to show the individual atoms. The colours represent different elements. Different portions of the DNA are called genes, and it's the genes which contain information about the physical makeup of the entire organism. In a human being, the information about the production of insulin is controlled by one specific gene. About 1 million people in Britain have diabetes, and that's an awful lot of insulin that must be produced to meet their needs. But why use bacteria to do that? Bacteria are among the simplest forms of life. Each one consists of a single cell with a single chromosome. They reproduce asexually. That means they don't have to have a partner to reproduce. When the cell is ready to reproduce, the single chromosome in the nucleus doubles up, so that when the cell does split into two, each of the new cells has a new chromosome, which is identical to the one in the parent cell. Bacteria can reproduce again and again like this, and with great frequency. Each generation is identical to the previous generation. That's what makes them so useful in genetic engineering. Scientists have identified the specific gene that controls the production of insulin in humans, and they've found a way of transplanting that gene into a bacterium. This is how it's done. As well as its single chromosome, the bacterium has other DNA, if you like, free-floating in the cell. This is called a plasmid. The plasmid is an easy target for manipulation. Using an enzyme as a chemical pair of scissors, the genetic engineer can cut out a section of the plasmid and put in its place the human gene which produces insulin. When that cell reproduces, the all-important gene is copied into the offspring again and again and again. Human insulin is an important product for drug companies. The genetically engineered bacteria are contained in these straws. They're transported in liquid nitrogen to keep them sterile. The straw is cut using a hot wire. The bacteria are spread onto a growth medium in a Petri dish. They need warmth to begin to reproduce. "24 hours later, they're taken out of the incubator. The bacteria have formed into small colonies. They're then put into a broth with different nutrients to grow on. Eventually, there are billions of them. They're put into a fermenter, and this is where the insulin is produced. This is the end product, clear, pure, human insulin, but made by bacteria. Genetic engineering of single-cell bacteria to make insulin is relatively straightforward these days. But what about the pea and the pig in our story? Is genetic engineering involving them as simple? You can bet it's not. In plants and animals which have more than one cell, it gets a lot more complicated. Any idea why? Well, one of the reasons is that in plants and animals there are different cells which have different functions. There are stem cells and leaf cells in plants, blood cells, bone cells, and nerve cells in animals. And both have cells which are specifically concerned with reproduction, sex cells. These are what the genetic engineer has to target. But that's nothing new. Farmers have been experimenting with the genetic makeup of plants and animals for centuries. To breed a cow with particular qualities, a breeder would select a male and female animal with those qualities and mate them. You've only got to look at how the shape of cattle has changed in the last 200 years to see that crossbreeding has been widely used. We could do the same with crops too, because they've changed over the same period. The problem with the traditional method of crossbreeding is that it's rather hit and miss. The results are not always what the breeder wants, and it can take a long time and many generations before the offspring show the desired qualities. The reason is that unlike bacteria cells, newborn sheep, cows, peas, wheat, and all the others are not genetically identical to their parents. This is because animals and most plants reproduce sexually. In sexual reproduction, half the chromosomes in the embryo cell come from one parent and half from the other. That's because cell division is different. When a cell is ready to divide, the chromosomes become shorter and thicker. They come together in pairs. The chromosomes in each pair pull away from each other and the cell begins dividing in two. The cell divides into two, each containing half the original number of chromosomes. These are the sex cells. Let's look at what happens in plants to illustrate the point. This is hand fertilization of a pea plant. The male sex cells, the pollen, of one plant are being transferred to the female part, the stigma of the other. Fertilized seeds will be formed, and new plants can be grown from them. The new plants will have half their chromosomes from the male parent and half from the female. They'll inherit a mixture of characteristics from both. Why does that complicate things for the genetic engineers? Well, they've got to ensure that any genes they want to introduce get into the sex cells of at least one of the parent plants they want to change. Let's look at an example. Imagine a food crop that would resist attack by insect pests and be able to fight off diseases all at the same time. This is a realizable goal for future years, and the first steps to achieving that have already been taken. Back to our pea. In this laboratory, plants are being bred which will be resistant to a toxic chemical, which is a type of weed killer. How is it being done? Part of the growing tip of the plant is to be treated. It's the most active area of the plant for cell division. A bacterium has been found which has a gene that provides resistance to the particular chemical. That gene can be removed using an enzyme. The DNA involved, which makes up the gene, is in the yellow solution. It's injected directly into the rapid growth area, the shoot. Some of the DNA will be absorbed into some of the cells, and some of those cells should become the sex cells. The injected shoots are put into a nutrient medium in a growth cabinet and left for a few days. Not all the cells accept the foreign DNA, so a small amount of the toxic chemical is put in with the growing shoots to kill off those that don't. So we've got young plants that have been altered by having the genetic material inserted. They're put into a carefully controlled environment to reach maturity. Those plants will then form the parent generation for a new strain of pea which will be resistant to the toxic chemical. Achieving this has much wider implications than simply producing super plants that resist toxic chemicals. Now that the technique for introducing genes into peas is known, many other more important characteristics will be worked on to see if they can be improved by genetic manipulation. That might involve, for instance, changing the amount of protein or carbohydrate in peas to make them more valuable as a food. Now to the pig. These pigs could help us to solve a serious problem, the shortage of suitable organs to be used in human transplants. Organs like the heart, kidneys and liver are virtually the same size in young adult pigs as in human beings. That makes them ideal for transplants. The problem at the moment is rejection. Because of the efficiency of the human immune system, an organ like a kidney transplanted from a pig would be rejected by the human host within minutes. Genetic engineering could provide the answer. Remember the plasmid that was used to insert the human gene into the bacterium? In a similar way, genetic scientists want to put a human gene into pigs to control the rejection process. But for now, they're having to use mice to perfect the technique. Being smaller, with a less complicated genetic map than a pig, the mice are easier to work on. They also reach maturity more quickly, so several generations can be studied in a short period of time. Scientists have succeeded in putting the genes connected with rejection into mouse cells and in producing offspring which carry the genes. But there's more work to do yet to achieve that with pigs. In the meantime, genetic engineering is raising many important questions. It's now possible, in theory, to transplant any gene from an organism into any other organism, be it bacterium, plant, animal or human. The benefits could be immense in solving problems of famine around the world, in finding new ways of fighting disease. Genetic engineering is regarded as one of the most exciting new areas of science. But it also means we have the potential to create organisms with entirely new characteristics, and it may not be easy to predict how they'll fit into existing ecosystems. Along the way, it may involve experiments with animals, which arouses controversy. There's the wider question of whether people should be interfering with nature at all. The genetic scientists would say that's been happening by methods like selective breeding for centuries. But then, they've never before had such powerful tools at their disposal. This is the story of a boy, a pea, a pig, and a lot of bacteria. James is diabetic. He has to do this 4 times a day. As well as his packed lunch, he has to carry with him this. It's a hypodermic needle, which he uses to give himself an injection. A geologist. He likes sports. Basketball is his favourite. And he enjoys weight training. It's only at lunchtime that there is anything different about James's day at school. The boy is James, and he's just like any 14-year-old turning up for another day at school.
There is a certain amount of...
James is in Year 10, studying for GCSEs and hoping to do well enough to go on to A-levels and university. He wants to be